So on this slide, I'm going to redraw the diagram that is written out or displayed on the previous slide so that we can go step by step through the life cycle of a virus. So let's start out drawing the cell membrane. And we'll do that in orange. So this virus is infecting a eukaryotic cell, and if you're unlucky, one of your cells. So here is the membrane. And just like in bacterial genomes, there are proteins and receptors that live in the membrane of eukaryotic cells. And I'm going to draw here a couple of receptor proteins. So there are some receptor proteins, and it's these particular proteins that the HPV virus attaches to and uses as a means to get into the cell. So the first thing I'm going to draw is the viral genome. So we'll draw the viral genome in purple. It's double-stranded DNA and it's circular. Not all viral genomes are circular, but this example is. So there is the circular genome, and of course all viruses have capsids, so we're going to draw the capsid proteins surrounding the circular genome. And notice that I'm drawing the capsid protein interacting with the receptor protein, and that is the mechanism by which this virus can enter into the cell. Okay, so the virus particle is able to bind to the receptor on the outside of the cell and uses that to enter into the cell. I'm also going to draw here the cell nucleus. So here's the nucleus of our cell. And after the virus gets into the cytoplasm of the cell, so here it is in the cytoplasm, the capsid proteins start to dissociate from the virus. So I'm going to draw kind of half or a handful of the capsid proteins to indicate that once the virus gets into the cytoplasm, then the capsid proteins start to go away and uncoat the DNA. The DNA then goes through a nuclear pore and makes it into the nucleus of the cell. So here we have the virus. Now, one of the goals, well, I shouldn't say one of the goals, the primary goal of the virus is to make lots of copies of itself. And once it's made lots of copies of itself, leave the host cell, and that way the different copies can go off and infect more cells. So one of the things that needs to happen in order to make lots of copies of itself is that you need to make copies of the genome. And this is done using DNA polymerase. Let's draw the DNA polymerase in green. Now, this particular virus uses host DNA polymerase. So it's using the DNA polymerase of the host cell to replicate its own genome. Now, there are some double-stranded DNA viruses that make or encode their own DNA polymerase, but in this particular example, HPV just uses the DNA polymerase of the host. So after the DNA polymerase does its job, you end up with multiple copies of the viral genome. Now, besides a genome, the cell needs other viral proteins like the capsid proteins in order to make new copies of itself. So what happens then, in that case, is not only are copies of the host genome, not only copies of the viral genome made, but transcription of viral proteins occur, again using host machinery. So what we have right here, or what I'm going to draw right here, is host RNA polymerase. So what does RNA polymerase do? Well, it performs transcription. So what happens is you end up with mRNA encoded by the viral genome. That's supposed to say mRNA. And since we're using host machinery, the mRNA gets transported outside of the nucleus, because that's what happens with host mRNAs, right? And in the outside of the nucleus, the mRNA encounters ribosomes. And what color should we do our ribosomes? Should we do our ribosomes in pink? So it uses host ribosomes, to synthesize viral proteins. So here we'll draw a little protein, the protein chain starting to emerge from the ribosome. And in this particular example, the key viral protein is the capsid protein. So here I'll draw pieces of the capsid protein. Guess I don't need to color them in. I didn't previously. So here you have capsid proteins that are then transported back into the nucleus. And now you have the components that are necessary to make new viral particles, and what needs to be done now is the new viral particles or virons need to be assembled. So here we have the poorly drawn new viral particle, and even though I'm only drawing one, there are lots. And it can now make its way outside of the host